So we are enter into that topic in a very small topic. Okay, we cut that topics into pieces. Okay, then only we can easily understand. Okay, so we start with genes. You can mute now. Okay. DNA. DNA, the chemical vehicle of heredity, that is composed of functional units, that namely genes. Okay, we know genes. What are genes? In DNA, DNA is the chemical vehicle of heredity. It is composed of some functional units. Functional unit of DNA is known as gene. Next, we are entering into another term which is known as genome. What is the genome? The term genome that refers to total genetic information contained in a cell. That term is known as genome. Genome of humans are more complex. We know 23 pairs of chromosome that contain 6 billion. 6 billion means 6 into 10 raised to 9 base pairs of DNA that contain almost 30,000 to 40,000 genes are there. Okay, so we discuss gene. Now we know what is genome. Then next we enter into the term gene expression. Okay, gene expression. Gene expression is the combined process of transcription of gene into mRNA, the processing of that mRNA and its translation into protein. We are very familiar with replication, transcription and translation. Then now we deal all these things in the terms of genome. Okay, gene expression. So gene expression, that is the combined process of transcription of gene into mRNA. The DNA contains gene that transcript into mRNA. Then the process of mRNA translated into protein. All these processes that contain gene, okay, that particular area is known as gene expression. Okay, then next you can see from this diagram, you know the chromatin remodeling, then transcription occurs. What happens in transcription? Premature mRNA is formed. We know RNA processing, yes, poly A tailing, methyl capping and all those things. Then we can synthesize this mature RNA by post-transcriptional modifications or RNA processing. Okay, then this mRNA leaving the nucleus, then enter into cytoplasm. Then this mRNA have stability. mRNA stability is provided by 5' methyl cap and also poly A tail. Then what happens? This mRNA enter into translation and synthesis of polypeptide occurs. Okay, we are very familiar with all these steps. Then what happens to this polypeptide? That undergo post-translational mechanisms and forms an active protein. In all those areas, we have genes. Okay, this gene expression, that last sentence, gene expression is the combined process of transcription of gene into mRNA, the processing of that mRNA and its translation into protein. Okay, Sandhya. Okay, Sandhya, you can unmute now. Okay. Yes, Sandhya.
Yeah, yes.
You have any doubt? Clear with these three terms: genes, genomes, and gene expression?
Yes, clear.
Can you explain what are genes?
Genes, the DNA, the chemical, the chemical vehicle of heredity. That is, in chromosome, one part of it is called the genes.
Okay.
Genome,
what are genomes?
The total genetic information in the cell. Okay. That is genome. That is, it contains 6 billion base pairs.
Okay, in human, almost 30,000 to 40,000 genes are there. Okay, yeah. Then what is gene expression? What is meant by gene expression?
Both combined process of transcription and translation. That is, transcription of gene into mRNA and mRNA to protein.
Okay, that complete process contains genes and that is known as gene expression. Okay, which are the 7 steps? You can just revise those 7 steps we discuss in molecular biology. Which are the 7 steps?
Chromatin remodeling, then transcription, RNA processing, then leaving the nucleus, mRNA stability, translation, and post-translational modification.
Can you name some post-translational modification? Do you remember?
Methylation.
Yes. Hydroxylation, carboxylation.
Hydroxylation, carboxylation, yes, ma'am.
Okay. What about RNA processing or post-transcriptional modifications? Surprise test about molecular biology. Okay. Okay. Any from the diagram itself, Sandhya?
Yes, ma'am. Yes, ma'am.
Foliate tailing and
foliate tailing, yes, ma'am. Capping and tailing.
Capping and tail. 3 prime, 5 prime, cap and 3 prime, foliate tailing.
Yes,
okay, okay. Okay. Happy to hear your answers. Okay. You remember all those points. Okay.
Yes.
Then, okay. Then you can mute now. Synthesis of protein under the influence is called gene expression. All genes of the cells are not expressed at all the time. For example, the insulin gene, it is expressed only in the beta, we know alpha cells, beta cells, delta cells of pancreas. The insulin gene is expressed only in the beta cells of pancreas, but not in other tissues. In other words, insulin gene is in a state of repression. There are 2 words, expression and repression. Okay. So, in other words, insulin gene is in a state of repression in all other cells. Then we are entering into 2 words, induction and repression. Induction is turning on the switch of the gene, and repression is turning off the gene expression. Okay. So, the synthesis of protein, we have to synthesize that particular protein when it is required. So, the synthesis of protein under influence. Okay. There is a sometime we don't want that protein. So, at that time we have to stop that synthesis, protein synthesis. In particular time we need that protein. So, at that time we have to start the synthesis. So, the synthesis of protein under the influence is called gene expression. All genes of cells are not expressed at all time. That is the more important thing. For example, the insulin gene is expressed only in the beta cells of pancreas, but not in other tissue. In other words, insulin gene is in a state of repression in all other cells. Okay. Then the term induction and repression. Induction turning on, repression is turning off. Then there are 2 types of regulation. Before that, Sandhya, you can unmute.
Yes, ma'am.
Okay. What is meant by turning on and turning off gene expression?
Turning on gene expression means for it to be expressed at all times. Like, for example, the insulin gene, it expressed only when beta cells of pancreas, but not in other tissues. Or in insulin is state of this thing. Turning on means the gene should be
expression. That means turning on. Okay, that a gene is expressed. What is meant by turning off? Turning off means
not allowing that expression of the gene.
That particular word is known as repression.
Repression.
Okay. Now we are entering into some terminology of genetics. Yes, okay. Expression and repression. Yes, turning on is expression, turning off is repression. Yes. Okay. In all cells there may be insulin, but it is not expressed, it is repressed. Okay. Yes. Okay. Okay, Sandhya, you can mute now. Next, we are entering into types of gene regulation. Regulation of genes are essential for the growth, development, and differentiation. There are 2 types of regulation, that is positive regulation and negative regulation. Okay. So, regulation of expression of genes are essential for the growth and development and differentiation. two types of regulations are there, positive regulation and negative regulation. What is meant by positive regulation? The gene regulation is said to be positive when its expression is increased by a regulatory element. Keen attention is required. Small topics, okay? So, what is this positive regulation? The gene regulation is said to be positive only when the expression is increased by a particular element that is known as positive regulator. The element or molecule that mediating positive regulation is called as activator or inducer. Simple thing, but it should be understood clearly. Okay, the element or molecule mediating positive regulation is called activator or inducer. Then one more type, that is known as negative regulation, a decrease in gene expression. Okay, this process is very important. Increase in gene expression and decrease in gene expression. Then this decrease in gene expression occurs due to the presence of another regulatory element. That particular regulatory element is known as negative regulator, and that particular process is known as negative regulation. So, element mediating this negative regulation is said to be negative regulator, silencer or repressor. We already use this term at the time of transcription or translation. I think enhancer, silencer, repressor, activator, inducer genes are there. But at that time, we don't know what is it. Okay. Now we know positive regulation, negative regulation, positive regulators and negative regulators. Okay. Okay, Sandhya, you can unmute now.
Yes, ma'am.
Okay. Can you give me examples for gene expression? We already discussed insulin.
Yes, ma'am.
So no need to explain once again. What are the types of regulation?
Positive regulation and negative regulation.
What is meant by positive regulation?
A positive regulation when expression is increased by some regulatory elements. That is called positive regulators that increase the expression.
Okay, they are also known as positive regulators are also known as
activator or inducer.
Okay, what about negative regulation?
They decrease the expression, or they have in presence of negative regulator, they don't allow the expression to happen. Example, they are called as regulators or silencers or repressors.
Regulators, silencers or repressors. In next topic, we are using these terms, okay? Yes. Activator, inducer, repressor and expression, repression. So you must know the definition.
Yes.
That's why I ask you questions again and again, okay? Then next, okay, Sandhya, you can mute. Next is about types of genes. Which are the different types of genes present in DNA? Okay, so the inducible gene that contain our positive regulator. Which one is known as positive regulator, or what is the name of positive regulator? Inducer or activator. So the gene which contain inducer or activator is known as inducible gene. So that is expressed only when a specific positive regulatory substance present. Okay, for example, beta-galactosidase. Beta-galactosidase is induced only in the presence of lactose in prokaryote. Where there is lactose, then only we have to digest, we have to split that lactose with the enzyme beta-galactosidase. Okay, insulin is an inducer of glucokinase of glycolysis. Insulin that decreases the level of glucose. So, in presence of insulin acts as an inducer gene for glucokinase. We know what is glucokinase, hexokinase. These are the enzymes present in glycolysis. So, when the glucose level is increased, insulin secretion occurs. Insulin acts as an inducer or activator. Then what happens? Then the gene expression of glucokinase occurs and the synthesis of glucokinase occurs. Okay. Then next is tryptophan pyrrolase. Okay, that is an enzyme that is involved in tryptophan metabolism. So tryptophan pyrrolase of liver that is induced. The synthesis of tryptophan pyrrolase is essential only in the presence of tryptophan. So tryptophan is known as the inducer of tryptophan pyrrolase. Okay, that is known as inducible gene. Then next is constitutive genes, which are known as constitutive genes. Genes whose expression is not regulated. Positive regulation and negative regulation is required only for this inducible gene. Okay. In the case of constitutive gene, there is no need of regulation. They are expressed at a constant rate, expressed always in all cells. That's why they are referred to as housekeeping genes. Okay. They are the they are present in cell always at a constant rate. Okay, as a result of mutation, some inducible gene producers become constitutively expressed. Okay, for example, the examples for constitutive gene that is synthesized by all cells and present at a constant rate, they are example as enzymes of TCA cycle. Enzymes of TCA cycle. What is TCA cycle? That is final common oxidative pathway for energy purpose. So we need TCA cycle. So the example enzymes of TCA cycle are synthesized by all cells. They are constitutive genes. Okay. Okay. Then one more category that is known as pseudogenes. Along with this inducible gene, constitutive genes, one more category is there that is known as pseudogene, that is used to represent DNA sequences that have significant homology to a functional gene, but they cannot express due to mutation. The pseudogenes are non-functional. Actually, they are not required. They are non-functional. What is the role of pseudogene? They increase the size of genome without any contribution to the expression of gene. Okay, they increase the size of genome without any contribution to the expression of genes. That's about our types of gene. Okay, Sandhya, you can unmute now.
Yes, ma'am.
Okay, we discuss the topic types of gene.
Yes, ma'am.
Which are the three types?
Inducible genes, constitutive genes, pseudogenes.
Then what is constitutive gene?
It's also called as housekeeping genes.
Why?
Because they are expressed at a constant rate and always expressed in the cell. As a result of mutation, some inducible genes produces and becomes constitutively expressed.
Okay. What example?
TCA cycle are synthesized by all cells. Every
Not
cell
TCA
requires.
cycle, not TCA cycle. It's a metabolism. The enzymes.
Sorry.
Okay.
Enzymes
All
in
enzymes
TCA.
are proteins. All enzymes are proteins, and these proteins are synthesized by replication, transcription, translation, protein synthesis. Okay.
Yes, ma'am.
Okay. We need that genes for the synthesis. Yes or no?
Yes, ma'am.
Okay. Then, which is the final common oxidative pathway?
TCA cycle.
What is the purpose of TCA cycle?
Convert everything to carbon dioxide and
Carbon dioxide and
energy and water.
carbon dioxide and water. Okay, okay. Then, which is the first material? Just I enter into metabolism. What is the starting material of TCA cycle?
Acetyl-CoA.
Acetyl-CoA combines with
Oxaloacetic acid.
Okay. That's enough. Okay. I just ask you what is TCA cycle. Okay. Then next is we must remember our carbohydrate metabolism, lipid metabolism. That's why. Then next is inducible gene. What is inducible gene?
They're expressed only when specific positive substance like an inducer is present. That is regulated positive regulation.
Example?
Tryptophan pyrrolase of liver. That is induced by tryptophan.
Okay. Then?
Beta-galactosidase induced by in presence of lactose.
What is lactose?
Lactose is that sugar, this thing, ma'am. Carbohydrate.
Yes. When we classify monosaccharide, disaccharide, oligosaccharide, which one? Lactose.
Large sugar or oligosaccharide.
Ah. What is monosaccharide?
Monosaccharide means it contains only one this thing. Sugars are only like that cannot be hydrolyzed to simple sugars. That is
Okay, that contain only one sugar acid. Okay,
yes.
saccharide means sugar. So
Yes.
monosaccharide only one sugar. Disaccharide,
2 sugars.
what is oligosaccharide?
Many.
Many, no, 3 to 10.
3 to 10. Yes, ma'am.
What is polysaccharide?
After more than 10.
Okay, very good. That is the classification of carbohydrate based on the number of sugar unit. Okay, so give me the examples for monosaccharide.
This
Glucose.
glucose. Then fructose.
Yes.
Galactose.
Okay.
Uh,
Then
yes.
galactose, mannose. Okay. Then give me the example for disaccharides.
Sucrose.
Sucrose contain 2 units. 2 sugar units are there. Which are they? Sucrose, table sugar.
Table sugar. Yes, ma'am. Yes, ma'am. It's a this thing. What is that? Glucose and fructose.
Okay. What about maltose? Maltose.
Maltose. I think 2 sugar.
2 sugar units. Which are they? Glucose plus glucose.
Okay, 2 glucose. Yes, ma'am. 2 glucose.
What about lactose?
Lactose, glucose, and galactose.
So lactose is an oligosaccharide or disaccharide?
Disaccharide.
Disaccharide. What are the contents?
Glucose and galactose.
So which enzyme is known as inducible? Which one is induced in presence of lactose? Name that enzyme.
Beta galactosidase.
Now it is clear.
Yes.
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 What is lactose? What are the contents of lactose? Which enzyme is used? Normally, lactose is digested by which enzyme? Lactase. Okay. Lactase. That is present in our intestine. Lactose splits into glucose plus galactose.
Yes.
Okay. That lactase present in intestinal brush border cells. Okay. Here, in this case, inducible gene concept beta galactosidase enzyme. Okay. Beta galactosidase enzyme is expressed only in the presence of lactose. Okay.
Yes.
We are entering into lactoperon concept. That's why I am stressed in this topic. Okay.
Yeah.
Okay.
Yes,
okay. No need to. Why, ma'am? Talk about this carbohydrate digestion and absorption. I don't want this. No problem for Sandhya. Can I enter into all areas? No problem. Are you okay with that? I can control. That's why. I can stick on this particular topic. I am always entering into all areas. That's why. Okay. Then next is what is pseudogenes?
It is they are non-functional. They are used to represent DNA sequence that have significant homology to a functional gene, but they cannot express due mutations.
What is the role of that pseudogene?
They increase the size of genome without any.
Okay. Without any purpose. Okay. Like our dietary fibers. Dietary fiber increases the bulk, then feels a satiety feeling, but it has some beneficial effects. Okay. Dietary fibers have some beneficial effect, but the pseudogene that increases the size of genome, similar nature and all those things, but they have no function.
Yes, ma'am.
Okay. Okay. I think now it is clear which are the types of gene: inducible gene, constitutive genes, and pseudogenes.
Yes.
Okay. Any doubt?
No.
No. Okay. You can mute now. Then next is operon. What is operon? Operon, we discussed this operon concept in prokaryotes, like our replication, transcription and translation. These are similar, almost similar in eukaryotes. Okay, so operon is the coordinated unit of genetic expression in bacteria. The concept of operon was introduced by Jacob and Monod in 1961. They got Nobel Prize in 1965 for this operon concept, and based on their observation on the regulation of lactose metabolism in E. coli. This is popularly known as lac operon. Lac operon. So operon, it is a coordinated unit of genetic expression in bacteria. Okay, this is popularly known as lac operon. Okay, then what is operon structure? We are entering into lac operon structure. Operon, it's a unit of genetic expression, gene expression, that contain so many elements. Which are the different elements present in this operon? That contain structural gene, control elements, regulator or inhibitor gene, promoter and operator areas. Then, which are the structural gene in this lac operon? ZYA. Operator gene O, promoter site P, then the regulator or inhibitor gene is I. Okay, so you just understand it's a unit. Operon is a unit of gene expression that contain different areas. Okay, that contains structural gene, control elements like regulator or inhibitor gene, promoter and operator areas. Okay, so what are structural gene? ZYA. In bacterial cell, Z gene. Now we discuss about lac operon. So Z gene that encode beta galactosidase. Okay, that enzyme hydrolyze lactose to glucose and galactose. Then next Y gene is there. Y gene is responsible for the production of permease. Okay, permease, that term indicates permeability, which transport lactose and galactose into the cell. Then XYA, A gene that code for thiogalactoside transacetylase. It's a lengthy enzyme. It's not a simple enzyme. Thiogalactoside transacetylase. Specifically, there is no significant role in this enzyme, but nowadays we find out detoxification of compounds that transported by permease. Along with lactose and galactose, some products are entered into the cell that is detoxified by this thiogalactoside transacetylase. Okay, these three structural genes, Z, A, Y, that code for the structure of the protein. These genes are present as a contiguous segments of DNA. It's present as a continuous segment. Okay, continuous segments of DNA. The transcription of these genes start from a common promoter. For the transcription of these genes, we need a promoter site that located close to the Z gene. Okay, the RNA polymerase binds to the promoter and transcribes these three structural genes as a single mRNA. You can see from this diagram, we can see that ZYA gene. Okay, here is a promoter site. This promoter site, to this promoter site's RNAP. RNAP means RNA polymerase binds and start the synthesis, promoter and operator. Okay, this is the structural gene and lac operon. Okay, Sandhya.